Hi, everybody. My name is Matt Boitano. I am the product manager for both the Revio and Vega systems, and I'll be your host of today's webinar. I want to welcome everyone to this webinar with today's topic focused on SPRQ-Nx and the first glance into SPRQ-Nx performance in the hands of our two beta customers. I'm joined today by Katy Munson and Maggie Gibson. Katy's from the University of Washington and Maggie Gibson is from Children's Mercy. Both will speak to their experiences with SPRQ-Nx and how they will apply SPRQ-Nx and have applied SPRQ-Nx in their labs. We have a lot of great material to cover today. The presentations are pre-recorded, but will be followed by a live Q&A session. You're welcome to submit questions at any point during the webinar by typing them into the area provided on the attendee control panel, and we'll get to as many of the questions as we can during the Q&A session at the end of the webinar. We've also uploaded some complementary literature in your control panel, so feel free to download those and take it with you. We'll also be recording this video and making it available for download sometime in the next few weeks. So please keep an eye out for a follow-up email with that information, as well as some additional information. Immediately following the webinar, you'll receive a brief questionnaire. So please do take a little bit of time to fill that out because it really helps us understand your needs and plan for future webinars. You can stay up to date on upcoming and future webinars for PacBio by visiting our events page at pacb.com/events. And with that, let's go ahead and get started. Okay, I'm going to start by making a quick introduction to SPRQ-Nx and describe a bit about the workflow and how it can improve your lab's workflows, projects, and economics. Since the introduction of HiFi in 2019, we've been seeing how our users are taking advantage of HiFi, how quickly they're adopting it, and how its ease of use, superior data quality, and unique sequencing characteristics are more confidently helping our customers and users achieve their research goals. From small projects like the Vega system as a learning tool in undergraduate and graduate level courses at the University of Miami, Ohio, to larger projects like single gene tests on individuals from varied genomics or population genetic studies like all of us. Our users are intentionally integrating HiFi into their labs to better serve the genomics community, and PacBio is continuing to make HiFi more accessible and a richer data type with more multi-omic capabilities. Right now, HiFi data is more accessible than it ever has been. The Vega and Revio systems offer unique advantages for our customers and our users to access HiFi data in a way that best fits their needs. The Vega system is a benchtop platform with an emphasis on ease of use and accessibility, perfect for that smaller lab that wants to take advantage of HiFi and the benefits that HiFi data provides, all in-house and under their own control. The Revio system is our HiFi at scale platform. It's capable of generating up to 2,500 human genomes per year and supports large-scale projects in carrier screening, population genetics, and clinical research. At PacBio, we strive to continuously improve our systems and consumables, and we'll be releasing updates in 2026 for both the Vega and Revio systems. For Vega, we'll be adding support for SPRQ chemistry, which will allow the reduction of DNA input requirements and bring chemistry parity to all of our long-read systems. We'll also be introducing short movie times for single-shot sequencing runs, 5-HMC calling on instrument, and support for 21 CFR Part 11 compliance. For the Revio system, we'll be strengthening our multi-omic offering with the addition of 5-HMC calls performed on instrument with every run, just like on the Vega. And just like 5MC and 6MA, which we currently offer on both systems, this additional capability won't require any additional library prep or treatment prior to sequencing, and therefore has no additional cost to the customer or the user. And we'll also be introducing a new sequencing chemistry called SPRQ-Nx, which enables the ability to reuse SMRT cells on the Revio system. This is what I'm going to be talking about a little bit more today. Both of these updates are geared towards driving value to our customers and enabling our users with the most up-to-date capabilities to help them drive home their research goals. With SPRQ-Nx, our customers will access complete genomes at a completely new price. SPRQ-Nx will bring the per genome price down by 30 to 40% relative to where we are today, and 10x down from where we were in 2022, making it the most affordable long-read genome ever. SPRQ-Nx uses the same library prep, the same instrument touchpoints, and the same data formats with no hardware changes as customers are using today, making it simple and straightforward to integrate smoothly into users' labs with the same but more sustainable workflow. SPRQ-Nx will look to make SPRQ-Nx broadly available to all customers in mid-2026. A little bit more on the workflow here. The SPRQ-Nx workflow is simple and introduces no new manual steps. Tracking and handling of SMRT cells is all automated and performed by the Revio system and the SmartLink software, so it's outside of what the user needs to think about and maintain. Run setup is identical today, just simply with less SMRT cells, since the system will reuse the SMRT cells multiple times. This is the key point that's driving down per sample costs and making a more sustainable workflow for our users. Each sequencing acquisition can be considered an individual event with different samples and applications for each SMRT cell use. There's minimal sample carryover, automatic washing performed on the instrument by the instrument, and the SMRT cells stay on the Revio system while being all while being managed by software. The automatic washing workflow is robust and removes nearly all previously sequenced library. Our customer speakers are going to touch on this a little bit as well, but sample carryover between uses is negligible and can be completely mitigated by use of different barcodes from use to use. In our internal testing data, we saw about 1 in every 10,000 reads carry over from one use to the subsequent use. And like I mentioned, our customer speakers will touch on what they saw with their beta runs and with respect to carryover. When we evaluate pre-development HG002 data generated at PacBio, we see consistent primary sequencing metrics across all uses, as well as excellent and consistent variant calling performance. Performance is similar, if not better, than today with Spark chemistry on the Revio system, allowing users to achieve their research goals more reliably and at the lowest cost per sample ever. These data, along with a series of plant and animal SPRQ-Nx datasets, are available for download and evaluation on our datasets page at pacb.com/datasets. Using those same HG002 datasets, we also wanted to look at how SPRQ-Nx data performs for secondary analysis workflows like genome assembly. Here we show that HG002 assemblies perform consistently across uses when looking at assembly completeness and contiguity. We are able to confidently deliver complete and accurate assemblies, including phased haplotypes with SPRQ-Nx chemistry. Again, if you'd like to download these datasets and evaluate on your own, they can be found at PacBio's datasets website at pacb.com/datasets. Finally, with SPRQ-Nx, we are increasing our multi-omic offering and adding the ability to detect 5 HMC base modifications in every run. 5-HMC is a lesser known and lesser understood base modification that correlates with multiple biological processes and has been shown to be alternatively present in some tissues and phenotypes. With HiFi data, we detect 5-HMC along with 5MC and 6MA via real-time monitoring of polymerase kinetics while sequencing. Base modifications change the polymerase kinetics, and we can detect specific modifications based on those observed changes. Adding 5-HMC detection alongside our current offering of 5MC and 6MA strengthens our HiFi multi-omic capabilities and opens new applications to HiFi sequencing, all while not requiring any additional steps, treatments, or cost to the customer or user and providing strong performance. We're currently running our SPRQ-Nx beta program and are extremely encouraged by the results and performance to date. Margaret and Katy will share their experiences with SPRQ-Nx beta next, but here I'd like to highlight the global beta performance. We have 4 external customers participating in SPRQ-Nx beta and have seen more than 300 total acquisitions as of January 23rd, when I collected this data. Average HiFi yields are around 125 gigabases per acquisition and are exceeding our spec, and customers are achieving greater than 120 gigs across all library types, while performance across uses is maintained. Read quality and base quality are similar or better compared to our on-market SPRQ chemistry under the Revio system, and the software and workflow experience have been reported as simple and robust. From here, we'll be opening up pre-commercial access globally in Q1, and then marching towards our commercial release in mid-2026. To learn more about SPRQ-Nx and how you can stay informed, visit our website at pacb.com/Revio. Thanks, and from here, I would like to throw it over to our customer speakers, Katy and Margaret.
My name is Margaret Gibson from Children's Mercy Hospital in Kansas City, Missouri, and we have been excited to test the beta configuration of the Multiuse SMRT Cells. I'll share a bit of information about HiFi sequencing at CMH, as well as our experience with the beta testing. First, a little bit of background on Children's Mercy. We were founded in 1897, and we are now one of the largest pediatric research and healthcare organizations in the Midwest. We have over 8,000 employees, 800 faculty members, 3,000 nurses, and over 1,000 allied health employees. We support a broad portfolio of diagnostic and research sequencing, which has provided the foundation for our adoption of long-read sequencing technologies in both research and clinical settings. In-house genetics and genomic testing at CMH operates in two separate but connected laboratories. The Clinical Genetics and Genomic Laboratory covers cytogenetics, molecular genetics, microarray, and next-generation sequencing and analysis and reporting, and performs both constitutional and oncology testing. Our overall clinical testing is about 90% constitutional and 10% oncology. We consistently see a 10% increase in our constitutional NGS-based volume each year, and that increase is without planned additions such as upcoming hospital expansions and internal expansion of testing. We estimate that we are still missing a fairly significant number of patients that should receive genetic testing, so continually do internal outreach to attempt to catch those patients. In particular, clinical PacBio sequencing has been growing quickly. We began offering HiFi whole genome sequencing as a laboratory-developed test in October 2023 and had 36 cases over the last 3 months of that year, which grew to 230 in 2024 and 290 in 2025. We have plans for further expansion as we continue to train analysts and clinical directors in HiFi analysis. The Genomics Core Laboratory is housed in the Children's Mercy Research Institute, also known as CMRI, and performs all of the NGS wet lab work for the hospital, including for the clinical lab and for any research projects. Our lab is CLIA/CAP certified and supports both internal and external clients. Overall, in 2025, we processed 1,500 samples for PacBio long-read genome sequencing, with nearly 600 LDTs and over 900 research samples. If you're wondering why the case and sample throughput don't match, each case has an average of 2.3 samples, as we most often run trios, kiddo, mom, and dad, which is why we ran more samples than cases in 2025. Within the Genome Core, we've been fortunate to be well supported with advanced instrumentation. Our core is equipped with Hamilton liquid handlers that streamline sample prep, 5 PacBio Revio instruments for sequencing, and a full set of quality control instruments. This existing automation and infrastructure has allowed us to scale quickly as demand for HiFi sequencing continues to grow. At CMH, research continues to be the main driver of innovation within our lab. Large-scale projects have provided the throughput, data, and operational experience needed to introduce emerging technologies like long-read sequencing into clinical practice. One of our main research projects has been our Genomic Answers for Kids, or GA4K program, which has been central to our research and discovery work. We recruit families, primarily those with unresolved or partially diagnosed rare diseases, and generate deep genomic data sets to support diagnosis and discovery. There are two main drivers of GA4K. First, to increase diagnoses in rare disease, and second, to develop a repository of genomic data that can be used for larger studies. Individuals enrolled in GA4K have a variety of sequencing and functional analysis completed, including tissue analysis, RNA sequencing, and third-generation sequencing, which at CMH is PacBio HiFi. GA4K is a long-term effort to build one of the largest pediatric rare disease genomic repositories. At last update, we had over 17,000 individuals enrolled and had made over 2,000 diagnoses within the study. the study primarily draws from the geographic area served by CMH, but we have participants from across the US and internationally. The QR code here links to our public information page for families and clinicians. Of note, our experience with HiFi sequencing in this cohort has directly informed our clinical adoption. Everyone here is probably aware of the benefits of PacBio over conventional genetic testing methods, but highlighted is a few of the reasons why legacy methods fall short. Karyotyping and microarrays can't detect smaller structural variants. Tandem repeat or STR testing is labor-intensive and often imprecise. Short-read sequencing struggles in repeat and pseudogene regions and for complex variants. And correctly aligning reads in areas of homologous pseudogenes is a compounding limitation of short-read sequencing. These limitations motivated us to explore long-read sequencing clinically as a more comprehensive solution. CMH has been performing next-generation sequencing for almost 15 years. In 2011, CMH established the Center for Pediatric Genomic Medicine and started a repository of research exomes and genomes. We launched our first clinical NGS test in 2013 with a 571 pediatric disease gene panel. In 2015, we launched short-read genomes, followed quickly by exomes in 2016. 2019 saw copy number variation detection from specific targets in short-read NGS testing, as well as the beginning of HiFi research testing at CMH, starting on the Sequel II instruments. To date, we have over 3,000 HiFi genomes completed on research samples. We built on that research experience and dataset to launch HiFi as an LDT in 2023, and that platform has expanded steadily as we validated its benefits. In our hands, we've seen HiFi increase diagnostic yield by more than 10% compared with short-read exome and genome sequencing. HiFi can detect all major variant classes, including those usually missed by short-read sequencing. HiFi testing has enabled correct diagnoses in cases that previously have been tested multiple times without resolution. And HiFi offers a more comprehensive approach to genetic analysis than legacy testing. Combined, these lead to improved patient outcomes. A review of our first 87 HiFi solves showed that 18% were only explainable with HiFi, meaning other legacy testing methods would not have fully explained these cases. We saw advantages across structural variants, methylation, phasing, repeat expansions, and pseudogene affected regions. While most cases would have been detectable by standard methods, HiFi brought clarity and in a few cases allowed dual diagnoses. In a 2025 paper, we compared the first 235 long-read sequencing patients we completed with 512 age and phenotype matched controls who underwent standard of care testing. Comparing our long-read sequencing cohort to standard of care testing, we observed a higher diagnostic yield, 37 versus 26%, an increase of 10% in the HiFi cohort. Most of this diagnostic gain was attributed to variants not routinely diagnosed by short-read sequencing. We saw a shorter average turnaround time. HiFi is faster by an average of 36 days, a decrease in turnaround time of 50%, and fewer tests required per diagnosis. The standard of care group focused on sequential testing to attain a diagnosis, while HiFi more often only required confirmatory testing. This reflects the efficiency of the single comprehensive test versus multiple sequential tests. Long-read sequencing clearly provides major benefits such as increased diagnostic yield. As mentioned, we see a 10% increase in diagnostic yield internally. Detection of complex variants such as structural variation, repeat expansions, and pseudogene resolution. Resolution of diagnostic oddities. Expanded diagnostic yields lead to the resolution of previously undiagnosed cases. Improved patient outcomes. More comprehensive genetic diagnoses allow better informed treatment and disease management strategies. Long-read sequencing is transforming rare disease diagnostics by providing a more complete picture of the genome, ultimately enabling better patient care, which is a central mission of our institution. Despite the numerous benefits, several challenges remain, impeding widespread adoption of HiFi sequencing. First, long-read sequencing is comparably high cost when transitioning from short-read technologies or other legacy testing. There are increased needs in instrumentation, data management, analysis, and storage requirements. HiFi requires specialized workforce training, both in the wet lab and for analysts. and there are regulatory hurdles such as CPT code development. All of these are highly relevant as we consider scaling long-read sequencing more broadly. The multi-use SMRT cells, SPRQ-Nx, have the potential to help mitigate the first of these challenges, cost, by decreasing cost per cell, which leads to decreased cost per sample. SPRQ-Nx allows more than one use of each SMRT cell, 2 uses per cell in the beta test, with independent samples on each use, which could significantly reduce per cell costs. We tested this technology using human whole genome samples from our standard blood DNA isolations. This is the same extraction we use on our clinical samples, though the beta samples are from research programs. Initial results are very encouraging, and we hope to transition all PacBio sequencing to the multi-use format. The workflow is very similar to standard SPRQ sequencing, but uses only one cell tray of 4 cells per 8 samples. There's a slightly higher risk of sample loss if a cell fails, since more samples depend on a single tray. Manual recovery is possible depending on the failure type, but increases the risk of sample mix-ups and hands-on time. The beta software had some minor issues, but nothing that blocked operation. Comparing standard SPRQ to SPRQ-Nx over the same time period, SPRQ had a 2.7% failure rate across 328 cells, while SPRQ-Nx had a nearly identical 2.8% failure rate across 72 cells. Very encouraging for a beta chemistry and means no new failure types were seen in our initial testing. Looking at key metrics for standard versus beta reagents, we compared HiFi yield, mean read length, quality, and loading metrics. SPRQ-Nx performed comparably or even slightly better in some categories. These results indicate that multi-use cells do not substantially compromise data quality. We also evaluated the first and second uses of the multi-use cells. Both uses had identical failure rates, 1 failure each. This suggests that second use performance remains stable with no new or increased failures. Looking at the same key metrics for use 1 versus use 2, use 1 produced slightly higher yield, while use 2 showed slightly longer mean read lengths. Both uses maintained strong quality values and consistent loading, supporting the feasibility of the multi-use workflow. Moving forward, we plan to transition all research samples to SPRQ-Nx and will run a full clinical validation once it's commercially released. The hope is to transfer all samples to SPRQ-Nx, as lower cost will help us provide PacBio sequencing to more people. We'd also like to see plate configurations supporting loading 16 samples at once to reduce hands-on time, which is significant when loading all 5 Revios at once. Ideally, uses would stretch beyond 2 uses per SMRT cell to further reduce hands-on time, increase efficiency, and hopefully reduce costs even more. If uses per cell increase, it would be best to see the sample loading configuration and loading process scale with the uses per cell. Overall, we are anxiously awaiting the full commercial release, as well as any enhancements that may come with that configuration. There's a large team that obviously contributing to this work. We need to acknowledge our teams across the genomic core, clinical genetics and genomics, genomic medicine, GA4K, and informatics, without which this work would not have been possible. And most importantly, our participating family and referring providers. Thank you.
So good morning, good afternoon, good evening. Thank you for asking me to come and present today on some of the results that we're getting from the SPRQ-Nx chemistry here at the University of Washington. My name is Katy Munson, and I'm director of the Long Reads Sequencing Center here at UW. A little bit about us here at the Long Read Sequencing Center. We are part of the School of Medicine, the Department of Genome Sciences. We are a PacBio certified service provider, and we are partially funded through the Howard Hughes Medical Institute. Our website is down here at the bottom of the slide. We'd love to talk with you if you have some projects that you want to discuss with us. We've been running the Revio platform since March of 2023. And over the last 3 years, we've seen almost 2,000 SMRT cells come through. The vast majority of those are for investigators here at the UW. However, about 17% are from outside investigators. And if we break down those SMRT cells into the kinds of libraries that we're running here, the vast majority of them are genomic HiFi libraries. With some Kinnex libraries, most of those are full-length transcriptome products, as well as some single cell, and then some amplicon libraries. We've been involved in several large sequencing consortia, including the Human Pangenome Reference Consortium, the Human Structural Variation Consortium, the T2T Consortium, who's interested in making high-quality telomere-to-telomere reference assemblies for humans and multiple non-human species, as well as the SMRT Consortium studying somatic mosaicism in human tissues. But some of the sequencing we're most proud of are the genomes we've been creating for non-human primates, including 6 great ape species that were recently published here in Nature. The project I'm going to talk to you today, however, is one of our involvements with the 1000 Genomes Project Long-Read Sequencing Consortium. So this group is trying to get high coverage long-read sequencing for all of the samples in the 1000 Genomes Project. Now, the 1000 Genomes Project is somewhat of a misnomer. It is a collection of human samples collected from around the world, and there's over 2,000 samples in the core collection, as well as over 3,000 in the full collection. We're hoping that by sequencing this diverse set of human samples using long reads, that we can identify a broader spectrum of genomic variation than is attainable with short read sequencing, and that this will further improve our understanding of human genetic disease. Currently, the consortium has made progress on about 500 samples using the Oxford Nanopore platform. And today I'm going to talk to you about some of the initial sequencing efforts we're doing using PacBio Revio on about 100 samples. Many of these samples, including most of the PacBio sequence samples I'm going to tell you about, we've been collecting transcriptomics data using the Kinnex protocol as well. So there's some transcriptome information alongside these high-quality genomes. The other technology I'm going to be introducing a little bit here today is called Fiber-seq. This is a long-read assay looking at chromatin architecture. And the idea is that when the DNA is still packaged up inside the nucleus, you treat it with an exogenous methyltransferase, which tags adenosine residues with a methyl mark. Once you extract the DNA from the chromatin and submit it to long-read sequencing, those methylated bases can be read out alongside the standard genomic information for each read. And this gives you a stencil of the chromatin architecture in a single molecule setting. So here on the right, I'm showing some example data from a Fiber-seq assay. These long gray bars are indicative of single molecule sequencing reads, with the locations of the methylated adenosines marked in purple. In general, what we see are regions of nucleosome footprinting that are marked by repetitive patterns of this adenine mark. However, here at the promoters of these genes, we see more extended regions of accessibility. And these are validated by orthogonal technologies such as DNA sequencing. So let's move on to the actual sequencing results that we obtained for this SPRQ-Nx beta program. The experimental design for this setup was to select 250 samples from the 1000 Genomes Project. We wanted a small overlap with some existing samples so we can compare them. The breakdown of these samples from the 1000 genomes is shown here on the left with the 5 continental supergroups. And then the bar graphs are showing in different colors each of the populations from the 1000 genomes collection. We ordered these cell lines, and a graduate student in Danny Miller's lab cultured them. We extracted fresh, high-quality, high molecular weight DNA in order to sequence these up to 30-fold coverage. For this experiment, we randomly divided the first about 100 samples between the Long Read Sequencing Center and our collaborators downstairs at the Northwest Genomics Center. Our protocols for library prep are fairly similar between the two sites. However, there are some slight differences. Specifically, at the Long Read Sequencing Center, we were piloting a semi-automated library prep using the HiFi Prep Kit 96 on a robot. Downstairs at the Northwest Genomics Center, all of the preps were done manually using the SmartBell Prep Kit version 3. The other difference is that we sequenced our libraries on the SPRQ-Nx beta chemistry, whereas Northwest Genomics Center sequenced theirs on standard SPRQ chemistry. Because of some of the limitations reusing these SMRT cells, it did make a change in terms of how we designed our sequencing runs from week to week. For SPRQ-Nx, we want to run the second use of a SMRT cell within a close amount of time to the first use. So on Mondays and Fridays, we would run 4 SMRT cells with 2 uses each, whereas on Wednesdays, we would run a single plate, typically with SPRQ chemistry for some other project. For our 1,000 genomes libraries, we first grouped them by size and then pooled 4-plex at equal molar ratios. We ran 3 SMRT cells for each pool and then checked on the total yield we got for each library. If a library didn't make it up to 30x genome coverage, we then requeued that for top-off sequencing. Because we're only running 3 SMRT cells per pool, we find that we have to run a different pool on a second use sometimes. So for example, pool 2 in the second SMRT cell is being run with a different set of barcodes. We always add the barcodes from the first use of the SMRT cell to the barcode list of the second use. And this allows us to see if there's any carryover of library from the first use to the second. For some of the fiber-seq data I'll be presenting later on, we didn't have this problem because we were running typically even numbers of acquisitions per library pool. However, we did have some instances where we'd run a Kinnex library just on a single acquisition. So to get to the actual data, I'm showing some raw sequencing statistics for SPRQ chemistry in the green dots compared to SPRQ-Nx in the pink. What we observe is a higher yield in terms of the total number of gigabase pairs we're getting off from SPRQ-Nx, associated with a higher QV score for SPRQ-Nx chemistry. Now, this is despite the fact that our libraries are slightly different sizes. So the SPRQ libraries are shorter and the SPRQ-Nx libraries happen to be longer. However, in the case of a longer library, we would generally expect a lower QV score, and here we're seeing a higher QV score. We think this is probably due to the fact that the raw polymerase read lengths are longer in the SPRQ-Nx condition, which is making up for the fact that our libraries are also longer. We see an increase in the total number of HiFi reads that reach at least Q20, as well as the percentage of bases that are Q30 or higher. Overall, for this project, we were able to sequence 50 libraries on SPRQ chemistry and 65 libraries on SPRQ-Nx chemistry using roughly the same number of SMRT cells or acquisitions in terms of SPRQ-Nx. The mean coverage of these data sets is fairly similar, although again, due to this slight difference in library sizes, the read lengths are different across the 2 conditions. The sequencing time was approximately the same for both sites. We expect that the project cost for this project is reduced by about 1/3 because we reused these SMRT cells for a second acquisition. Although because we were able to sequence more libraries on the same number of acquisitions, there could be further reductions in cost if this yield increase holds. Our next step with this project is to actually dig into these data sets. We want to compare them across analysis such as de novo assembly and variant calling for structural variants, tandem repeats, and so forth. So here are the same data sets that I just showed, comparing SPRQ and SPRQ-Nx on standard HiFi libraries. However, now I'm going to add some more data points for fiber-seq tagged libraries using both SPRQ and SPRQ-Nx sequencing. Our SPRQ chemistry libraries in this case are historical data over the past year or so, so they are certainly more dispersed in terms of yield and read length. However, the same trends hold in terms of the fiber-seq libraries. So SPRQ-Nx is showing us a higher yield and a higher QV score across the board. Again, this is probably due to the increase in the polymerase read length, and we see an increase in the number of reads as well as the quality scores of the bases. Digging a little bit into this data, here I'm showing the fiber-seq tagged data for several ENCODE cell lines, where the fiber-seq tagging density is in the purple histogram, and we're showing CpG methylation in the brown histogram below the bars. This is an example gene, and we see, as we expect, that chromatin accessibility at the promoter of the gene is high. However, across these different cell lines, we see different footprints across the body of the gene. And this is expected because these cell lines are derived from various human tissues and would have, or from various tissues and would have different chromatin accessibility. If we dig down deeper into the data, so on the right side are the same kind of individual lines of reads with the purple tags showing the methylated adenosine. And the same nucleosome footprints can be observed, as well as the peaks of accessibility. Overall, we are seeing no difference in the data between the fiber-seq tagging of the SPRQ-Nx libraries compared to standard SPRQ chemistry. I also took a look at the data between the second use of a SMRT cell compared to its first use. Specifically here, I'm only looking at cells where the same library pool was used for both first and second acquisitions. What I'm noticing is that the yield from the second use is certainly typically not less than the first use and potentially even greater than the first use. Also, if we look at the libraries within those pools, we see that there is a slight but significant increase in the read lengths that we're obtaining on the second use compared to the first use. If these trends hold, then that's a really good sign that we're not losing anything by reusing the SMRT cell. I also looked at the barcode usage for second uses in the case where a different library pool was used for the first use compared to the second. And overall, I'm seeing less than 0.02% of carryover of first use barcodes into that second use data set, which is fairly minimal and definitely something we can work with. So overall, we find that the SPRQ-Nx chemistry has a similar workflow to standard SPRQ, including the benefits of a reduced library input. However, there are certain additional workflow considerations. For example, the fact that the cells must be reused within a certain amount of time. And there may be some carryover of first use libraries into the second use. However, these are things that we can work around. And we see that the second use is showing comparable or slightly higher yields, as well as read lengths than the first use. For these high quality genomic DNA samples, we see equivalent or higher data yield and quality scores compared to SPRQ chemistry. This extends both to standard HiFi as well as fiber-seq libraries. I haven't shown you the data, but we've also run some Kinnex libraries on SPRQ-Nx, and we see similar performance on SPRQ-Nx compared to SPRQ. However, we do need to do some more additional testing to see if these are really working as well. Overall, we would like to test a much broader variety of sample types, for example, non-human samples or samples with much lower DNA quality. I'd like to thank the people that did this work, the technicians at the Long Read Sequencing Center, as well as our collaborators in the Miller Lab and Strigachis Lab, and our collaborators at the Northwest Genomics Center here at UW. I also want to thank our contacts at PacBio for giving us the opportunity to test out this new chemistry. And thank you for listening.
All right, thank you, Katy and Maggie. Now we can begin the Q&A portion of the webinar. We did have a couple of questions, handful of questions roll in. I'll start with a couple that are for PacBio, and then there's also a couple here for Katy and Maggie. The first one is there a plan to bring SPRQ-Nx to the Vega in the future? With the Vega update that we'll be making in 2026, we will be bringing chemistry parity to both Revio and Vega. So they will be having the same sequencing chemistry. The Vega system just initially won't have the ability to use SMRT cells multiple times, but it will contain the same sequencing chemistry. The second question is regarding the cell wash and reuse. At what point in the run is the cell washed? Does a first use cell get washed at the end of a run, then returned to a tray and used for the next run when it started, or is that cell washed need to be reused within the same run? So the cell is washed at the end of data collection. So after the end of that specific cell has been sequenced, it is then stays on that instrument. The instrument takes care of all of the washing. The wash buffers are contained in the sequencing plate. So the instrument just pulls those buffers from the sequencing plate and washes on the work deck of the instrument. That SMRT cell can then either be used immediately following that wash, or there can be a gap in order to set up your next sample and then start your subsequent run or second use of that sample, subsequent use of that sample. This is a question specifically for Maggie. Margaret, have you done cost analysis on HiFi genomes versus standard of care that includes labor? I'm curious about time spent doing analysis or manual review of variant calls and interpretation for HiFi versus short reads and ancillary assays.
Yeah, so that is something that we are working on. Obviously, insurance companies and other payers are also interested in that because they're more likely to pay for a test if we prove it's cost effective. We're still gathering the information. And so as people gain experience, their time per analysis will come down. So the math is going to change there. And then we also obviously expect the math to change with the decrease in cost for PacBio genomes on a per sample basis. So in progress, but we don't have data yet.
Great, thank you, Maggie. Another question is, how long does the automatic wash take between runs? It's fairly quick, on the order of 10 to 15 minutes, so relatively quickly. Is the polymerase kit changing? What about the sequencing plate? The polymerase kit will not change for SPRQ-Nx. The polymerase kit will not change for SPRQ-Nx, but the sequencing plate will be updated with SPRQ-Nx chemistry, as well as the buffers that are used for washing in between uses. Were the dataset comparisons all based off single sample per SMRT cell? Katy, Maggie, did you all pool and sequence multiple samples within a SMRT cell, or were they all single plex?
So my data were mostly multiplex. Our standard was 4 samples, 4 barcoded libraries per SMRT cell per acquisition. So yeah, the barcoding worked all the same as a standard SPRQ run.
We routinely don't pool samples. We're running 1 sample per because we need that level of coverage for our genomes.
Great, thank you. Sifting through some of the questions. Here's one for both of you. How easy was it to integrate the SPRQ-Nx workflow into your existing Revio lab?
It was shockingly easy, honestly. We expected more issues than we had. So it was a great experience for us.
Yeah, for us, it was just a question of timing. So we typically run 4 cell plates a week, which doesn't divide by 2 very well. But all in all, the transitioning into the next workflow was very, very straightforward.
Cool. Another question here. Does it, I'm going to paraphrase, but essentially the question is, can you run different libraries on different uses of the same SMRT cell? And the answer to that is yes. You can run independent libraries, independent applications. So each acquisition should be considered just an independent acquisition. And I'm assuming Katy and Maggie, that's typically how you are running in the data right now.
Yeah, the one trick is because the Kinnex barcodes are different, that can cause a little bit of a workflow change. But the data look fine.
Okay. We've only run genomes so far.
This question is specifically for Katy. You mentioned higher accuracy with SPRQ-Nx despite longer inserts. Did you see any impact on analysis results?
So we don't have that data yet. These are all pretty hot off the presses, and we're still working on the analysis results. So stay tuned. We're hoping to have those coming out pretty soon.
Okay, cool. Let's see. This is another one I think for both of you. Can you share a bit, looking ahead, what types of projects do you think SPRQ-Nx unlocks? And will you be transitioning the bulk of your sequencing, HiFi sequencing, to SPRQ-Nx?
Yeah, we plan to transition everything to SPRQ-Nx, and I'm not sure it will unlock new avenues, as we are pretty focused in pediatric rare disease and undiagnosed disease, and so I think it will just enable us to offer that for more people.
So on our side, we're mostly a research lab, and we do all sorts of projects. What we envision for next is the ability to do larger cohorts. So to be able to kind of get into some disease studies with long reads, because we know that those reads are accessing different areas of the genome than short reads. And just the price hasn't been at the point where we can do larger cohorts of samples. So we're really hoping to be able to attract those kinds of projects based on the cost per sample coming down.
Great. Here's another one, I think probably for both of you as well. Are there any specific library prep workflows or, sorry, I'm paraphrasing this again, but workflows or steps that need to be changed or adjusted or optimized when switching from SPRQ to SPRQ-Nx?
Not for us. We prep samples that went on both chemistries on the same batch of samples. So it was exactly the same sample prep ahead of the actual loading of sequencing.
Yeah, we have made no changes to our library prep. We've even taken the same bind and loaded it on both SPRQ and SPRQ-Nx, and it works just fine.
I don't see any additional questions coming in. Are there any maybe last comments from Maggie or Katy before we close?
I mean, like Maggie said, in our hands, this beta worked out really well. And we are looking forward to the official release and being able to offer this more broadly to all of our customers. And as we go through, we are starting to do more testing on some of those things with the lower quality samples. And as far as I can tell at this point, the performance is really very, very similar / even better on Nex. So I'm very excited about this and about, again, just integrating some of these little quirks into the new workflow. I don't think it's going to be a problem.
Yeah, similar thoughts here. We're excited to transition everything to it when it is commercially available.
Okay, thank you both for that. Actually, I think maybe my rallying call elicited some more questions. So we do have a couple more. Has pure target have pure target libraries been tested or validated on SPRQ-Nx chemistry? We will validate and test all supported applications prior to commercial release, but not all applications were run during this beta phase. I'm sorry. My questions keep jumping. Given the results of reusing the SMRT Cell, is PacBio planning to test the upper limits of reuse in the future? We'll always continue to try to push the boundaries, but we always want to make something that's reliable and consistent performance in our customer hands. Do reagents in the sequencing plate remain exactly the same except the wash buffers? No, that's a good question. The sequencing chemistry that comes on that sequencing plate has been optimized with this release. So we should see some important performance consistency improvements with the SPRQ-Nx chemistry. Maybe Katy, this one is for you, since you specifically called out read length differences. Any ideas about read length differences from SPRQ to SPRQ-Nx, even with the same shearing method?
So I have an N of 1 that just came off yesterday. So that was before we recorded the webinars. this change that we're seeing in terms of the second use being just slightly longer, I took one library and I ran it on Spark and on SPRQ-Nx. This was that same bind on both platforms. And we see, again, that very, very slight, less than a kb difference in mean, where SPRQ-Nx is showing actually just a little bit shorter on that first use. Second use should be coming out in about 12 hours. So I'm hoping that that second use is going to bring it up. Now, that being said, it's really, really slight. And because we're getting more data off the SMRT cell, we actually get more data at all read lengths. So, you know, if you're worried, that doesn't seem to be a problem. However, again, we're using Pippin size-selected libraries, and we don't have any bead wash-based libraries on either chemistry at this point. So that may make a more of a difference. But for the libraries that we run, we're not seeing any significant changes in read length N of 1.
Okay, thank you. Another quick one. Has anyone seen an increase in mitochondrial DNA coverage? That's not something PacBio has specifically looked at, but we do have, like I mentioned earlier in my talk, we have SPRQ-Nx and Spark HU002 datasets available at our datasets page at pacb.com/datasets. That's, if you're interested, you can download those datasets and do some self-evaluation if you'd like. Another one, is there an update to SmartLink to enable SPRQ-Nx and SMRT Cell multi-use? Yes, there is. There's a SmartLink update as well as a system software update to support the new chemistry and the new workflows. Okay, I think that might have been it. If there are any additional questions that roll in, we'll make sure to follow up either with the speakers or PacBio and follow up with that whoever asked that question to provide an answer. But with that, I'd like to thank Katy and Maggie for their time and dedication to this webinar. We really appreciate you coming on to speak about your experiences and expertise. And I'd also like to thank all of the attendees for your time, your attention, and taking time out of your day to listen to us. Just as a reminder, as soon as the webinar closes, there will be a brief questionnaire. If you could fill that out, it really helps us plan and optimize these webinars for your benefit in the future. But with that, thank you again for joining. Hope to see you all on future webinars, and I hope you have a great day. Thank you.